Hi, my name is Mia, and I'm going to be talking about the resting, local, and action potentials. For the resting membrane potential, it is a bunch of neurons that exist across the plasma membrane and reflect on the ionic differences between the cytoplasmic and extracellular sides of the membrane. The leakage channels, which are these two, this one is sodium and this one is potassium. So the non-gated ion channels in sodium potassium help pump to maintain the polarized state. So we have sodium, which is one of the dominant cations of the extracellular fluid, while potassium is the dominant cation of the intercellular fluid. The presence of the leakage channels in the membrane also allow these ions to diffuse across the plasma membrane in direct opposition in the movements through the ions and of the ions, which is sodium and potassium. So sodium is only going to go this way and potassium is only going to go this way. For the local potentials, this is what I have drawn. And there's actually two types. The first type is the excitatory postsynaptic potential, and that one will occur in the local region of the cell membrane, and that would be depolarized. So for the depolarizing ones, it will occur in response to the opening of the specific gated ion channels in the membrane that result in a network of sodium into the cell, which causes the localized region of depolarization on the membrane. There's another one, but I did not draw it. And the second type is like an inhibitory postsynaptic potential, and that takes place when the local region of the cell membrane is hyperpolarized. For the hyperpolarized, the opening is either a ligand-gated potassium or chloride channels, depending on the specific neurotransmitter. It allows either the potassium or chloride that would both result in a localization, hyperpolarization of the membrane. For action potential, I drew the neuron. So basically, I was kind of hard to draw, but think of it as like a light bulb just traveling, and it can go at different rates. So the action potential is like more of like a dramatic change, and it's produced by voltage-gated ions channels in the plasma membrane. It occurs only when there's a high enough density of voltage-gated channels. Most of the body has only 50 to 75 channels per square micrometer. It's mainly just called the trigger zone. So there are temporal disseminations that occur. So as there are multiple signals that are sent from the same presynaptic terminal in a rapid-fire fashion, the reduced time between the signals result in an increasing strength of developing that potential graded potential. And once it reaches the axon hillock, that is also called the trigger zone. This is kind of like what's going on. So once that threshold, like the rapid fire, where it's like fast enough for it to actually trigger, we have the sodium channels with the trigger zone open, which I drew this. It's like this originally would be kind of closed unless that trigger zone was triggered, basically, which results in an influx of sodium ions that cause depolarization and begin the propagation of AP, which is action potential.